Yeah. So my talk is titled Reconstruction of the Synaptic Venn diagram, which I also call connectome of the fruit fly brain. So in my research, my goal is to understand how neural circuits are organized and how they perform complex computations and ultimately give rise to behaviors. And that means that we first have to really create maps of those brains. And the biggest map, and the one I will be talking about, is the map of a whole Drosophila brain. This was a huge consortium effort that also broke through the mainstream media last year. And in this talk, I will tell you the approaches that enabled this resource, and more importantly, why it matters and what we can do now that we have a map of all the individual neurons that you can see in colors here and the synapses between them. And that really starts with the fruit fly itself. I assume most of you are familiar with fruit flies, especially in your kitchen. Fruit flies are also amazing model organisms for science. There are 6 Nobel Prizes that were given for research in Drosophila, including the circadian rhythm was found in Drosophila. So it's really been remarkable. And another benefit of it is that compared to other model organisms, such as mouse, it has a fairly small brain. So with 139,000 neurons, we can now map circuits that span the entire brain. And despite its small brain, Drosophila has a wide variety of complex behaviors that are well studied and described. And it has a wider range of sophisticated senses, such as it can taste with its feet, and it can, of course, also hear, smell, and see with those big eyes. So one big question is how these senses are integrated in the brain and give rise to these complex behaviors. And a lot can be learned and has been learned from measuring individual neurons, like the activity of them in the brain. But now that we have an entire map of the brain, we can now trace circuits, pathways from the inputs to the outputs. And Drosophila was not the first animal that has its brain mapped. The first one was actually C. elegans. You might have heard of it. It has 302 neurons. This brain was mapped back in the 80s. And this is what the connectome of it looks like. So we can represent graphs or connectomes as matrices. On the one axis here, we have the presynaptic neurons. On the y-axis, we have the postsynaptic neurons. Every dot is a connection. And we see here there are 2 types of connections. There are chemical and electrical synapses. And in this talk, I will actually only focus on the chemical synapses. The ones that we think are mostly important, but I want to highlight there are other synapses as well. So Drosophila has been a model organism that was early adopted for the creation of connectomes due to its size. And the first ones were created, especially in the visual system. The visual system is very repetitive. So the idea was if you do a small part of it, you know about everything. That was followed up by work on a part of the brain that was called the hemibrain, 25,000 neurons. And that was really the first resource that had large parts of any brain in it and led to many analyses. And then including here at UW, this work was done at Geneva, but this work was done at UW here. We also now have a ventral nerve cord, which is essentially the spinal cord of the fruit fly, reconstructed. So that is in the Tata lab just over in the next building. So what we did was going beyond that and reconstructing the whole brain with 140,000 neurons and all its synapses. And all these approaches, they start with high-resolution images of the brain. And electron microscopy has been the approach of choice so far because it allows us to image the brain at high resolution and at large scale. So this image here is a slice through the Drosophila brain. And because it's taken at high resolution, we can zoom in and see the individual details within it. So for instance, here we can see neurons that are outlined by membranes. We can see these blotches here, these are mitochondria. We can see these blobs here, these are vesicles that are filled with neurotransmitters, and they're found at synapses. So in this place here, we have a synapse where the darkening is, where this one neuron contacts these other neurons that have a red star in them. So with this data, we can find synapses, we can see individual neurons, but to make this an approach that scales to an entire brain, we have to make it 3D. And the way to do that is to slice the brain in many thin slices, 40 nanometers thick in this case, and collect them individually, image them, stack them back together to a 3D block. And if you do that successfully, you can then flip through the slices, and then if you latch onto one of those, you can follow a neuron as it traverses through the brain. So this way of mapping connectomes has been optimized dramatically over the last 10 years, and now we can trace it big enough that we can trace circuits that span the entire brain. So here's one neuron. These are all the synapses incoming and outgoing. This is just one neuron that it contacts with. And so there's one connection between those two neurons in the brain. It's just one of 140,000 neurons. So in this talk, I will tell you about cell reconstruction, how we go from these images to reconstruct those cells and find the synapses, to build a whole connectome. And then ultimately, I will talk a little bit about neuroscience and give you an idea of what we can do with a connectome. No neuroscience knowledge required, but I think you will see how this data is useful for analysis. So to start, we have to basically color in all those neurons. And it's like a big coloring book. You have to fill in the neurons with the same color. And that we call segmentation approach that has been dramatically optimized over the last years. And if you look at this and you think, well, that is just like segmenting objects in images, you're right, that is the same. However, I want to highlight this field has realized that this needs to be solved computationally early on. So in 2010 already, the field adopted convolutional neural networks before they were cool to segment images. And we can even go back to the C. elegans connectome. That was in the 80s. Actually, Sidney Brenner, he hired John White because he was a computational scientist. And in this paper, there's a quote that says, some use was made of a computer-aided reconstruction system described by White, that was his PhD thesis, and Stevens and White, but most of the reconstructions were done by hand from a total of about 8,000 brains. So they realized that we needed computation approaches, but the computers were not powerful enough back then. And that is part of the reason why the field kind of fell into a freeze between the C. elegans connectome and now the new connectomes that are coming out, because the computation power to actually reconstruct those data sets was not available. So we optimized segmentation dramatically, but for the same reasons that you're not using a self-driving car today, these segments are not perfect when they're being automatically generated. There's still errors left in the segmentation that have to be corrected. So here I'm showing you a segment that comes hot off the press, like an automatically reconstructed, not quite neuron. And we can see it's not a single neuron because there are, for instance, multiple cell bodies in it. And one neuron has one cell body. So we know something is wrong. There is, for instance, one error here where there's this one branch that runs horizontally and the one diagonally that have been falsely merged together. So there's an error. And if I flip back and forth, you can see how this single error here creates like a problem that makes this entire component not ready for analysis. And because just a few errors ruin an entire segment, virtually in a large data set, virtually no segment is correct. And we have to correct these errors through proofreading. So that's basically where I was at the beginning of my PhD. I wanted to analyze these data sets. I had to do proofreading, and no good tool was available to actually do proofreading of these data sets. And I had a choice to make, basically, how to implement proofreading. Usually, most projects in neuroscience are done that one person has one rig, one job, and creates one data set that maybe gets combined later, but usually there's one person doing the work. However, it was clear that this ultimately has to be an effort that is done by many people in parallel. And that means also to solve the challenges, the computation challenges, to allow a whole group of people to work together to correct and proofread these data sets. And that meant creating a new data structure that is hosted in the cloud that everybody can access. And I will tell you a little bit about more what is going on in the background here. And for that, we have to pull away the curtain a little bit and look at the segmentation. So on the left is segmentation like I showed you before. But what we actually do is we create first what we call an over-segmentation, where we paint voxels, 3D pixels, that we believe definitely belong to one object together. We call them supervoxels. And then we combine these supervoxels to the large voxels, to the large segments. So we generate a graph of supervoxels. Two supervoxels have an edge between them when they touch. And in this graph, the kinetic components are the neuron segments. So again, we actually generate a supervoxel segmentation where voxels that definitely belong to one segment are grouped together. And then we add edges between them so that we can run the kinetic components. And then the kinetic components in this graph are the neurons in our data set. And that means that proofreading can be described as operations on the graph, not on the actual voxel data. And so when we want to merge two things together, we insert an edge. And if we want to split things apart, we remove edges. The tricky thing here is actually that every time we make one of those operations, we have to recompute the kinetic components. And that is challenging in large data sets. And to highlight this, here is a naive implementation of this kinetic component algorithm, where we have all the supervoxels down here on the x-axis, and the ones that belong to one kinetic component, they just point to one neuron, an ID, you can imagine. The problem is that in those graphs, in the fly brain in particular, we have over 2 billion supervoxels and 10 times more edges. So if we have to recompute kinetic components all the time, that will take way too much time to do if somebody wants to interactively go through and correct errors. And just to highlight this, even just reading the edges from a big table, which is a very fast data structure, takes 10 seconds just for one neuron, even without computing the kinetic components. So this approach does not scale. So what we did is we married this kinetic component approach with the spatial structure of the data set. We implemented an octree structure. And here on the right, you can see one neuron and how it has all the boxes. These are little chunks. And in an octree, as you go higher in the hierarchy, the chunks get always double in size. So when you reach the top of the data set, you would have just one chunk for the entire data set. And the idea here is that if you make an edit, the edit is only very local. It's just in one little location. And that means that all the other chunks are not touched. So the idea is, can we reuse this information in the other chunks and just make one local change? So how is that turning out in our data structure? So here, this means that we insert an additional hierarchy where we group locally supervoxels together that belong to one kinetic component in one chunk. So we have a local kinetic component that points to this one node. And as we go up the hierarchy, we combine kinetic components together to larger and larger pieces until we reach the top, and then we have one neuron. So why is this beneficial? Let's look at a split. And the split, remind you, is removing edges. So we remove an edge down here. And instead of having to load the entire thing, we just have to load the within chunk graph. We computed a new kinetic components locally, create two nodes, and then we can reuse the kinetic components in the neighboring chunks. We don't have to ever look at them. We can just go up the hierarchy and combine our kinetic component. So this now scales with the log. It's a binary tree of the largest dimension of the data set, and it's vastly more performant compared to the naive approach. And here I'm showing you real evaluations from real user interactions. On the x-axis is the naive just for the reading of the edges. On the y is the hours for in this what we call a chunk graph approach. And with that, we were able to bring edit times to about a second. So now people can come in from anywhere in the world, connect to this data structure, and make edits with single-second times. And additionally, we built in time versioning and locking to allow many people in parallel to proofread and also to be able to go back in time to retrieve old states. So this is what a merge looks like. So here I'm finding the extension of this neuron. You can see this black is an artifact in the data. So here's an extension of this one neuron. I zoom in. I click on both sides to indicate a merge should be done here. And when I zoom out, the edit is already performed on the server. And critically, everybody who comes along and clicks on this neuron, they will see this new version of the data because we all work on the same backend structure. So this implementation now allows a large group of people to proofread in parallel. And here I'm just showing you 4 neurons that were proofread over time in this fly data set. So proofreading is really one piece of the puzzle, but we realized the second that you hand out the ability to proofread your neurons to a group of people, those people come back and they want to analyze the data because they are proofreading to get the circuit of their interest. And the problem here is the following, or the challenge, I should say. So this is a neuron before and after proofreading. So during this proofreading, segments were added and removed. So here I'm showing you in red those segments that were removed of this neuron, and in green the ones that were added. And the thing is that there are synapses attached to these segments. So by proofreading, not only do you add and remove segments, you also add and remove synapses. So you're changing the connectivity of the connectivity information in your data set through proofreading. And if you have, so here on the right, these dots here, individual, these are synapses that are attached to these fragments. And effectively, their association with the neuron were changed through proofreading. So you have now a group of people doing proofreading, making changes constantly to the data. You have another group of people wanting to analyze the data in a way that is reproducible. They want to be able to write a paper and put it out and have somebody else rerun the analysis and get the same result. And that is challenging. And here, I will not precisely tell you how we did that, but I want you to know that we created what is called the connectome annotation versioning engine that solves, that combines proofreading annotation and reproducible analysis queries in any connectomics data set. It is now used for over 20 of those data sets, especially the largest ones that are out there today. And that was a big collaboration between Allen and Princeton, and we have a team that has been working together on this for 7 years to maintain this kind of infrastructure for the field. So this is the backbone that allowed us then to launch what we call Flywire. Flywire used the whole brain data set from the fruit fly that I showed you before, an automated segmentation, and allowed any fly researchers to join and proofread the neurons that they were interested in. And not only proofread, but also analyze, because it was hosted in Cave. So the upshot was this. You can join, you proofread, it takes about 20 to 30 minutes per neuron to proofread, and you can proofread your neurons of interest, you can do your science, publish a paper, but in the process, you contribute to the proofreading of the entire data set. And the wild thing is that this was completely open. Anybody could see anybody's work. And where people were overlapping, they had to talk to each other about credit, and that worked remarkably well. So on the next slide, I will show you 3 years in one slide. Every dot here is an edit that was made in the data set. And by the end of it, we made about 3 million edits, manual edits together. And at first, here on the left, I'm showing you this over time as a rate of number of edits per day. And these bars show you what it effectively means per minute. At first, this was mainly people that joined to do their science. At some point, we realized we have a chance to finish a whole brain connectome, and we put in a lot of centralized proofreading, paid proofreading, that was hosted by Princeton and the LMB in Cambridge, UK. And together, we finished everything that the scientists didn't finish. But even during that time, we had people join, and this is the number of people making at least one edit per week, and we had even that number was increasing. So over time, we had over 200 people making at least 100 edits in this data set. And I want to really, and together we proofread all the 140,000 neurons with over 50 million synapses in this data set. And I really want to highlight that there were a lot of people that contributed to this resource. Most people through proofreading, but many also through contributing tools for analysis or for the reconstruction, the segmentation, and all that. So there are over 200 people, 280 people, over 50 institutions that contributed in some shape or form to this resource. And my most favorite part about this is probably that not only did people contribute, they were also able to do science. So these are 34 papers that used this data set before it was published. Because we made the data available, where things were already proofread, where circuits already proofread, people were able to do analysis and publish using this data set. Not only could they do analysis, we also had people contribute data For instance, the M data I showed you before, but also the synapses were actually contributed from the community. And I want to give you just a few examples. So here the synapses. The synapses are these little connectors between pre and post sites. These were created in the Funke lab that were added to create the connectome. And excitingly, also very importantly, the Funke lab also typed these synapses. So synapses, you may be aware, have a different effect depending on what type they are on the postsynaptic neuron. Most commonly, it would be excitatory and inhibitory. Do I excite my postsynaptic partner or do I inhibit it? There are also other ways how neurons affect each other. In this case, they looked at 6 different neurotransmitters, and they showed that just from looking at the high-resolution electron microscopy data, they can make a prediction about what neurotransmitter a synapse emits. And this was very crucial. They applied this to the entire connectome, and every synapse, every edge between 2 neurons now has a type. And we can say something about if there's an excitatory and inhibitory pathway, and we will use this in a second. And there were many, many annotations of neurons. You have to give your neurons names and types to understand who is involved in your analysis. And one approach we did was to collaborate with the Jeffries lab. Strong collaborators for the entire project and specifically for the typing. So here we did a hierarchical approach of typing all the neurons in the brain. Like first, we grouped all of those that are within the brain, intrinsic, and these come in 4 main groups. Those that are completely intrinsic to the optic lobes, like most of the neurons in this data set are actually optic. So the fly is a highly visual animal. Those that are in the central brain, here in the center, and those that project from the optic lobes into the central brain, and those that project from the central brain out back into the optic lobes. Then we have those neurons that project out of the brain. So those are some motor neurons, especially for the extension of the proboscis, that is the snout for feeding. And we have a couple of few endocrine neurons, and then we have descending neurons. Those are the main output pathways out of the brain. They run down the ventral nerve cord or the spinal cord and drive motor neurons for walking, flying, head moving, and so on. And lastly, we have those neurons that project into the brain, ascending neurons coming up from the ventral nerve cord, and sensory neurons, especially photoreceptors, as you can see here on the sides. So this data has everything we need to go from sensory input to this brain to output of the brain. And I will show you now one little example of how we can do that. And for that, we will turn to the ocelli. The ocelli are 3 simple eyes on top of the brain. The fly has technically 7 eyes. The 2 big ones, the 3 ocelli on top, and then 2 very small ones on each side that are mainly there to track light levels for circadian rhythm. So the 3 ocelli on top of the brain, they are very simple eyes. They're under focused lens. They will not do any object detection. They will mainly do brightness levels, and they're tuned to UV light. And they are in the photoreceptors, they reach into the brain, and we can actually see the axonal end of them that I'm showing you here, and we know how they exit. So we know from which eye each of those photoreceptors is coming. So per eye, we have 80 to 100 photoreceptors coming into the brain. And the question is, what do they do? Like, how do they contribute to the fly's function? And people have hypothesized before that they may be important for stable flight, and I will show you why we think that is indeed the case. So in the brain, so we know they come from left, medial, and right eye. So in the brain, to our luck, there is a region, the ocellular ganglion. It's a round region. It has glia sheets dividing it into 3 parts. So we know for every neuron in this region what photoreceptor it receives input from. And so we have the medial, the left, and the right photoreceptors would terminate in these separate regions. And the main output of this region are the ocellular ganglion cells. These are massive cells by comparison. And we're now looking at the brain from behind. There have been reported to be 12 of them. And here I'm showing you those colored by the expressed neurotransmitter, by the likely neurotransmitter. And those are cholinergic and glutamatergic. And we think cholinergic means excitatory in the fly brain, and glutamatergic means inhibitory in the fly brain. So here I will just use excitatory and inhibitory to talk about those neurons. You can also see that there are actually two descending neurons in brown here mixed into them. So that was not known before. So we have neurons that run directly from the eyes, receive photoreceptor input, and they run immediately down into the ventral nerve cord onto motor output, an extremely short circuit. So probably helping for reflexes. But it gets better. We can look at these from the top and look at what eyes these receive input from. So here I'm showing you, again, these three eyes, the regions, and each of these ocellular ganglion cells only receives input from one eye. So here I'm showing you one excitatory neuron on the left, and it receives input like in half of this region of the left eye. Here, this inhibitory neuron receives input from half of the region on the right eye. So we can assign information about from what eye each of those neurons receives input from. And what we found quite strikingly is that when you look at the neurons and you look at this excitatory neuron, it crosses over. So what turned out to be that the excitatory neurons, they project what's called ipsilaterally to the other hemisphere, whereas the inhibitory neurons, they stay on the same side. So now the question is, do they target the same downstream neurons? And the answer is they do. So here on the x-axis, I'm showing you the excitatory number of synapses on any downstream target as a dot. And the y-axis, I'm showing you the number of inhibitory synapses onto any downstream target. And you can see that there are a group of neurons, descending neurons, that receive strong input from both excitatory and inhibitory projection neurons. And this is, and we can summarize this like so, where we have inhibition that stays on the same side and excitation that crosses over to the other side. And that is akin to what has been described as a Breitenberg vehicle in the 80s. So in a Breitenberg vehicle, we have two sensors and two wheels. And the sensors and the wheels are connected for this specific vehicle, like so, similar to our diagram. And the sensor is driven more strongly the closer it is to a stimulus. So in this case here, this sensor would be driven more strongly, meaning this wheel is turning harder. And it means that this whole vehicle turns towards the stimulus. And for the inhibitory circuit, that works just the same way, just that it balances out the excitatory drive. So the closer this vehicle would get towards the stimulus, it would not run into the stimulus, it would slow down. And so this is showing you why we think that this circuit is helping the fly with stable flight by orienting it to a stimulus. These eyes are tuned to UV light, so when it's outside, that it can stay upright to the sun. There's much more to the story. Of course, I just showed you one slice of the ocella. As a whole, I only showed you one slice of the entire data set. And there are already many, many studies that use this data set for analysis and complete new directions for research coming out of it. Just want to give you three examples here. First, this is a study by Sapkar et al. They looked at neurons important for halting. So every time you see the red dot come on, the neurons on the right, for these two flies here, there is a specific neuron, a different neuron in each of them that is activated. And as you can see, the fly stops. So these neurons promote stopping of the fly. And what they found is by simulating the connectome locally is that these are two different kinds of halting. One is that the fly is using when it's feeding and it wants to just halt and to eat. And the other one is literally stopping while running. So there are two different mechanisms in the circuit that allow the fly to stop. And they could find that out through the connectome. A whole simulation of the entire data set has already been done by Xu et al. And one thing that how they tested that their simulation is actually correct is by looking at how taste neurons impact the specific motor neuron 9. So motor neuron 9 is important for the extension of the proboscis, so the snout, for feeding. And in their simulation, they activated neurons, taste neurons for sugar and for water, and tested how motor neuron 9 would respond in their simulation. And then they went to a different fly, not ours, a different fly, and they activated those neurons in that fly and measured the activity of model neuron 9, and they showed that there was good agreement between their simulation and what they found in this other fly. So, and lastly, we can now finally start comparing connectomes. The field has really been an N of 1 for a long time, like you have one connectome of one animal. Now we can at least compare ours with the one that I showed you before, the hemibrain. That's a part of the brain, but where they overlap, we can compare them. We can find the same neurons because the fly is so stereotypical. So you can see on the left here, this is one specific neuron type. And you can see on the right here how this looks like in our data set. And you can see how similar they look. So you can find the same neurons, then you can analyze the circuits that they're involved in and ask questions about how stereotypical are those brains, how much variation is there, biological and otherwise. And in this paper, Schlegel et al., they find high agreement between the two. The most important thing is this data set is publicly available for anybody to explore. Codex is an interactive resource that you can go in and type in the neurons of your interest. You can look for the ocellular ganglion cells and projection neurons and look those up and see the targets are the descending neurons. And yeah, and then to summarize, I told you about the whole brain connectome of the fruit fly. It was an open community effort that was enabled by new computation approaches. And in this case, I told you about CAVE, or in part at least about CAVE. And the ocellular circuit analysis is just one example how we can now map pathways from input to output and suggest new hypotheses about what these circuits actually do. Most importantly, the data is publicly available on Codex and also for download on Zenodo. So in the last few minutes, I just want to go beyond the fly brain and put it into context. So the field of connectomics has really been working very hard to scale this approach. Here on the x-axis is time over the last 10 years. On the y-axis is volume in terms of data set volume that was acquired with electron microscopy. The fruit fly brain is not the largest data set that we have today, but it is the largest one that has been fully proofed. CAVE has been used for multiple of them and is currently used for many, many more, upwards of 20, to reconstruct these data sets. And another example is a data set of a human cortex. It's a small part of a human brain, of course, but it's about a cubic millimeter of a human brain. And the largest data set today is this one here. That is a cubic millimeter of mouse visual cortex acquired by the Microns Consortium, consisting of Princeton, Allen, and Baylor College of Medicine. It is also publicly available and on micronsexplorer.org. And in this data set, which is about 40 times larger than the fly brain itself, we can reconstruct connections between individual cortical neurons. So here I'm showing you two pyramidal neurons, the dominant cell type in the cortex. These are excitatory neurons. And you can see that these have thick dendrites and very thin axons that run very far and wide. And this one here is an example of these two pyramidal neurons and one synapse between them up here. So here I'm showing you DEM. And that is probably one of the longest reconstructed connections between two neurons in a connectomics data set. Proofreading each of those takes about 50 hours for a single person. So really, we are at the limit of where proofreading scales. And that is the reason why in this data set, we have only about 1,700 neurons being fully proofread. So if you want to scale this approach to a whole mouse brain, you have to solve proofreading. And the mouse brain is about 20,000 times larger. Many of us are working towards this goal. And proofreading it right now would cost about $7 to $20 billion in human labor. Of course, we will not pay so much money for proofreading, so we have to automate proofreading. And that's one target that I'm personally working towards too. Okay, with that, I want to acknowledge all the amazing people that I got to work with in this project, especially on the Princeton side and in Cambridge, but also at the Allen Institute, and of course, the whole FlyAway consortium that came together in a completely new way, at least for connectomics and neuroscience, and made this possible. Yeah, and thank you for coming and looking forward to questions.
Thank you so much, Sven. That was great. Are there any questions or clarifying issues that Sven could address?
So is a data set like the data set online that we could access, that's from single fly or, okay,
A single female fly.
okay, and so there's plasticity, of course, in these systems. Is there any idea that that can be looked at in a reasonable time for, so for example, a young animal with an older animal?
Yeah, so plasticity, the changes of synapse sizes to activity, is something that in the fruit fly at least we know is happening in certain regions, but most of the connectome is actually quite stereotypical. So if you go to another fly and you find the same connections about the same strength, and that is what some of these comparative approaches are doing. The nice thing is that the fly has a few regions where you can actually look at this question and you can analyze that. So how age plays in, I don't know for Drosophila, because the larva stage is so different that most of the neurons are actually shed and removed and are not retained to the adult stage. That said, I think on the cortical side, for mouse connectomes, we can do that. And I actually have an analysis on plasticity in the mouse visual cortex. And there are ways to look at fingerprints of plasticity that show you, that allow you to make some estimates. I'm happy to chat more about that later.
I was just curious,
Yeah.
if like this manual proofreading, there's room for errors? And if there's errors, how is this impacting this whole?
Yeah. No, that is a very fair question. Yeah, so we have, I mean, it's basically Wikipedia style in some ways. That's how we can think about that. And we had a lot of checks in place of analyses, especially when you compare left and right, for instance, of the brain, that should lead similar results. The cell typing efforts were able to pull out some discrepancies that were then revisited. So you can do statistics on top of the whole connectome and to gain confidence that there are not many errors left.
I'm wondering, because you mentioned scaling this, and with like automatic methods to scale, there's going to be errors as well. And how much, like, what's the percentage of error you can live with without this affecting the understanding of how the brain works?
So I think ultimately, I always said the entire process, there's no such thing like a perfect data set. Like there's always something left to do. You will always find a little thing that is wrong. And in fact, this data set remains proofreadable. Like not the public version that is a static snapshot, but people that are in the consortium can still make changes. And we'll make a new version at some point available if there are enough changes in there. That said, it looks like that even a few errors don't impact accuracy much in the analysis. Most connections in the fly brain are very redundant. So most have tens of synapses. Losing a few does not impact the result much. And ultimately, there is the confidence in the process that the processes we designed in a way to be sure about the accuracy.
Synapse-associated glial cells, if they exist in fly, or you track that in mouse, other glial cells?
Yeah, glial cell types glial cells are segmented just like any other neuron in the data set, or any other cell for that matter. And they're glial cells in Drosophila. There was actually just a paper that came out last week on bioRxiv that analyzed glial cells. I haven't read it yet, but that also looked at our data set and looked at the glial cells. They are not well proofread, all of them, but we have many well reconstructed. And in the Drosophila, they look a little bit more like astrocytes. And I think that's like the main type that we have. But in the mouse data sets, we have beautiful glial cells of the main 4 types. And there is also an analysis on oligodendrocyte precursor cells, the OPCs, in a smaller data set. And I think these data sets are very valuable for this kind of analysis as well. And happy to chat more if you're interested in that.
Yeah, so a few slides ago, you had a graph showing like over time how the size of like data sets increasing. So do you expect that trend to like continue in the same fashion? And do you think your ability to like proofread will scale to that size?
Yeah. So, and I also did not actually say, I meant to say this is a log plot. The y-axis is in log scale. So this is exponential growth. Linear scales in this plot is exponential growth. And so I think what we will see is that on the large end, people will still push the limits, and we will see that scaling. Right now, the NIH is funding up to a scale up of 10 cubic millimeters, which would be 1 order of magnitude higher, which should be reached in about 4 years. So I think on the higher end, we will see some kind of scaling, and I think people are very confident that we can get to the mouse brain at about 500 cubic millimeters quite well. But I think we will see a different trend coming, which is that now that we have a whole Drosophila, there will be many studies that just want to do more of Drosophila or of the same organism and compare. And I think that's a very exciting direction. You can start looking at, I mean, of course, the next step will be comparing male, female, but you can start comparing different Drosophila that have certain conditions and understanding what is different on a circuit level. So I think you will see basically labs just cranking out Drosophila connectomes and making comparisons, and that being a whole research direction and a kind of scaling too.
I was wondering if we could speak to like how imaging technology changes, maybe like, you know, like if these images are 5 years old, you know, is that still as useful as maybe one that's taken today, as well as like yeah, I think artifacts that might be used.
So the funny thing is, in some ways, that these electron microscopes that are being used for this, they come from the 80s.
Yeah.
And so the technology for 2D has been around for quite some time. What is new is the productionization of the whole workflow to make the 3D work consistently and well, to be able to cut 20,000 times without introducing too many artifacts. And that is what is improving, and that what people have been pushing the limits of. And so what you would see is that the number of artifacts will go down as we get better, and then we push to higher limits of larger data sets that have still some artifacts left in them. So that is what you would see if you look at these data sets. That said, there's actually one technology that just came out last year that might be disruptive, and that is expansion microscopy. So electron microscopy has the benefit that it has higher resolution than light microscopy, of course. And light microscopy, you can't use that for connectomics. However, you can blow up your tissue 20 times or more, then you get, and then you can image with light microscope, and the resolution you get effectively can be now as low as what one can achieve with EM, or almost as low. So there was the first paper, the method is called Icon, where they showed that this is now possible to do. And many are currently reproducing that, and there's a whole frenzy and trying to figure out if that is the next thing. But any new method will start at the bottom. Like they will have a data set size of around down here. So it will take all this productionization, all that scaling over a decade to reach the same volumes that EM can achieve today.
The same lines like image segmentation techniques.
Yeah.
Are gonna, maybe perform better on the better imagery. So, and even on the bad imagery for that matter.
Yeah. So my view on segmentation is that most segmentation algorithms, they work on all of them work on the voxelized data. So they load in the raw data onto a GPU, and then they say what's inside and out. And the problem with that is that they don't have a context that we have when we do proofreading. Like when you do proofreading, you see that two things that shouldn't be connected, and that's why you make your decision. Mm-hmm. They don't have that context. They can only see a local cutout, and growing this cutout means that that's going to be the power of 3 ultimately, and you're limited by how much you can fit onto a GPU or a TPU. And that is why I don't think we will ever reach necessarily high performance level with this approach. Mm-hmm. So what I see happening is that instead of pushing this limit further, we will automate proofreading. So we have a second stage of a process that can, just as the human, look at the whole thing as a single segment instead of everything at once, and can find the errors and correct the errors. And I think that's where we will see the next leap in terms of scaling up and
type of approach, or what would be the second step?
I mean, I showed you like a very simple example of that earlier where we looked at this one here. Like we know three cell bodies shouldn't be in one object. So seeing this and then finding the place where these things are falsely connected, you can do when you see the whole thing. And that is just what we do, but ultimately when we train algorithms, they might see things that are more sophisticated or more black boxy than what we see. But I think this is the way how we will go about achieving the next step in performance.
Can I ask you a really like not data sciencey or sciencey question? Like, why is the fly brains not symmetrical? Like the fly brain, like there's one side bigger than the other.
No, you mean
yes.
this flap here? So this flap is an artifact.
Okay.
So it turns out that like when you prepare a fly or a brain for imaging, the photoreceptors are attached very, very thinly. So the person doing this, I mean, it was a miracle that the photoreceptors stayed on actually, most of them, but they came off a little bit on the lower left. So on this side, the reconstruction of the photoreceptors in particular is not as good as on the other side. So that is one thing. But we went into this not even thinking we were able to do the photoreceptors to begin with. So that was a win still. So the brain is actually extremely symmetric.
Okay.
So if you look into the brain, it is incredibly symmetric, and we have an analysis on that in the paper.
Thank you so much, Sven.
Thanks for having me.
Other questions? Yeah, please reach out to Sven later. Thank you so much for being here today. I'll see you next time.